growth is defined as an increase in the number of constituents in cellular structures. When there is growth in the absence of cell division, there is an increase in the size and weight of the cell. Whereas when growth is followed by cell division, there is an increase in the number of cells. It is important to distinguish between the growth of individual cells and the growth of populations, since microorganisms, due to their small size, do not do individual growth studies but population growth studies. Population growth is the increase in the number of cells as a result of individual growth and subsequent division. Population growth occurs exponentially. Exponential growth is a consequence of the fact that each cell divides, giving two daughter cells. This process is known as binary fission, which when divided will give each two daughter cells, so in each period of division the population doubles. Curve of bacterial growth. The figure illustrates a growth curve of a bacterial population. This curve is divided into 4 phases called latency phase, exponential phase or logarithmic phase, stationary phase and death phase. This phenomenon occurs only in a closed bioreactor or batch type. This type of closed bioreactor is characterized by changing the physiological and environmental conditions inside. In our case, primary metabolites are produced in the exponential phase and secondary in the latency phase. In this culture medium, there is no exit or entry of cells in culture medium. This type of bioreactor is easier to maintain sterility conditions and then extract cell mass and metabolites. It also allows to obtain the genus Bacillus thuringiensis in its sporulated and non-vegetative form, and also to obtain the Bacillus subtilis in its vegetative form with all its antibiotic substances if it is known when to cut the process in this type of bioreactor.